for joining today's Advances in Autism Research and Care webinar. Before we get started, I just want to review a few housekeeping items and let everyone know how you can participate in today's event. So right now we're looking at an example of the GoToWebinar attendee interface, which is made up of two parts. We have the viewer window on the left, which allows you to see everything the presenter will share on their screen, and the control panel at the right. And within the control panel is how you can participate in today's event. So it allows you to choose your audio mode, and during the presentation, all attendees will be muted. It also allows you to ask questions, either using the questions pane, or you can raise your hand and be unmuted to ask a question. We'll address questions and answers at the end of today's session. And just a quick reminder, today's webinar is being recorded, and everyone will receive an email with a link to view the recording, the slide presentation, and an evaluation form that can be completed for CME credit. And so now I'm going to pass the microphone over to Dr. Corey, our Network Medical Director, who will introduce today's presenter.
Great. Thanks very much, Audrey. I'm very pleased to have our presentation today on advances in the genetics of autism spectrum disorder. It's been a couple of years since we last visited this topic, and it's always a very popular one, and there are always advances taking place. Just about every week I read something new that's going on in the area of autism genetics. Our speaker today is Dr. Abha Gupta from the Department of Pediatrics and Child Studies Center at Yale. Dr. Gupta received her bachelor's degree in molecular biophysics and biochemistry from Yale, and then her MD PhD at the University of Pennsylvania. She completed a residency in general pediatrics and then a clinical fellowship in developmental behavioral pediatrics at Children's Hospital of Philadelphia and at Yale School of Medicine. Her main interest area is with autism spectrum disorders, and I think she has a wonderful presentation for us today. Dr. Gupta.
Thank you, and good afternoon. So I'm happy to talk to you about recent advances in the genetics of autism. And this is a topic I would say could be covered within a one-hour webinar a few years ago, but now it's impossible due to the explosion in research that's happened. So I hope to give you an update about the most important and interesting findings. And so to get started, I'll just say that I have no conflicts of interest to disclose associated with this program. And then I know there's a wide variety of backgrounds among the audience, so I thought I'd start off with just a brief review of Genetics 101 for those of you who haven't thought about or studied genetics in a long time. So the cells in our body contain a compartment called the nucleus, and that houses our genetic material, which consists of 23 pairs of chromosomes. So there's one pair inherited maternally and one pair inherited paternally. And so in this very simple diagram, it shows how our genomic material is organized in chromosomes. And what makes up these chromosomes is the DNA ladder or DNA double helix. I'm sure you've heard of that term. So the genetic code is made up of these rungs of the ladder, which consists of bases, or otherwise known as nucleotides. And these are chemicals that pair up in a very specific way, and it's the specific sequence of these bases or nucleotides that determine the code, the sequence of amino acids in our proteins. So that's why it's referred to as the genetic code. So when human geneticists look for mutations that might cause a disorder like autism, they look for things like sequence changes. So here again is a simple diagram of the DNA ladder with the base pairs making up the rungs of the ladder. And let's say there's one of these rungs is mutated in this example. This could be for a variety of reasons. It could be inherited from a parent. It could be spontaneous or de novo, meaning that it occurs in the germline. Or it could be caused by an environmental trigger, such as ionizing radiation, etc. So this mutation, this sequence change in the code could be benign. It might not cause a problem, or it could be really damaging and could cause something like autism. So human geneticists look for sequence changes to associate with a disorder, But there's another layer of complexity to the human genome, and that's copy number variants, or CNVs. So in this case, we usually think of having 2 copies of every gene. Again, one inherited from our mother, one inherited from our father, but it's actually not so simple. So for example, I could have 2 copies of a gene, someone in the audience could have 3 copies, someone else could have 1 copy, and we could all be perfectly fine. It might not cause a problem, but then there are some CNVs that can cause a disorder like autism. So in this diagram, again, pretty simplistic, you have an area of a chromosome that's duplicated, and here you have an area of a chromosome that broke away and became lost. And even though the rest of the chromosome came back together again, now you have deleted genetic material, which could affect really important brain active genes. So human geneticists look for sequence changes and copy number changes to try to associate with a disorder. Oh, there we go, next slide. So, whoops, let's go back. Okay, sorry about that. So there are different scales of genomic variation that human geneticists study. So on a chromosomal level, you could be looking for changes such as I showed before, duplications and deletion on the order of millions, hundreds of millions of base pairs. We can look for copy number variants, which are typically considered to span tens of thousands of base pairs to a few million. And then also at the single gene level, which is usually about 3,000 nucleotides or bases long, and look for sequence changes at the 1 base pair level. Next slide. Let's see. Okay. All right. So unfortunately, whoops, I'm afraid I'm jumping slides here for some reason. Let's see if we can get, okay. A little bit tricky here. Okay, here we go. Let's see if we can, all right. So there are several lines of evidence for a genetic basis of autism. Unfortunately, as recently as the 1970s, there was this thought that autism was due to bad parenting and this unfortunate term of refrigerator mothers. But fortunately, since then, the community has realized that there is a biological basis for autism. So there are several lines of evidence for genetics. One is sibling recurrence risk, so asking how often do you find a second child with autism in a family already affected by autism, and that number is close to 20%, which is much higher than what you would find in the general population, meaning that something is running in families. However, you could argue that what that is could be an environmental trigger causing the disorder. So human geneticists have tried to get around that by looking at twins. So looking at identical twins, which share 100% of their genetic information, and fraternal twins, which are like any brother-sister pair, they share about half their genetic information. So these twins you would expect to be growing up in roughly the same environment, but identical twins, the question is, would they share a diagnosis on the spectrum more frequently than fraternal twins? Because if that were so, then you would expect that autism was due to something genetic. And there are studies that have shown this, that identical twins do share a diagnosis or are more concordant than compared to fraternal twins. However, I can tell you that identical twins are not always concordant. In other words, 100% of the time they are not always sharing a diagnosis on the spectrum, although the number can get close up to a 90% in some of these studies. So any human geneticist in the field would admit to you that there are probably other reasons, other contributing factors for autism, including environmental triggers that we're unclear about. And then another line of evidence for genetic basis is the fact that about 10% or more of cases also have a defined genetic syndrome. So here's a table showing some of these autism-related genetic syndromes, and I'm sure you've heard of some of these, such as Fragile X and Rett syndrome and tuberous sclerosis. So many of these have been traced to mutations in specific genes. And if you look at the proportion of patients with the genetic syndrome that also meet full diagnostic criteria for autism, it can be quite high. So for example, in Fragile X syndrome, about a quarter of males meet criteria for autism and about 6% of females. However, if you look at the reverse, what proportion of patients with autism have that genetic syndrome, the numbers are quite small. So what this tells us is that these syndromes are rare causes of autism, and they're not necessary or sufficient to cause autism. So why are they important to know about? Well, for one thing, because a lot of these syndromes have been traced to specific genes that can give us some insight into what might be contributing to autism. So if we know which gene is involved, then we can figure out what that protein product does, where and when in the brain it might act, and kind of uncover the disease mechanism, the pathophysiology of autism. So I want to present the bottom line fairly upfront in this talk, and that autism spectrum disorders are complex. Everyone knows that. There's wide phenotypic variability. There's multiple etiological factors, and there's a great deal of genetic heterogeneity. So in other words, there is no one gene for autism, not even a few. In fact, recent genetic studies are estimating that there are likely hundreds, maybe up to 1,000 or more. So there are about 20,000 some genes in the human genome, and about 80% of those are active at some point in the lifetime in the brain. And so there are a lot of genes that can go and perhaps produce a common final pathway phenotype like autism. So how do human geneticists go about trying to identify genes associated with a disorder? There are two main approaches. One is the positional approach, which looks genome-wide and is unbiased, which makes no hypotheses about what chromosomal region or what gene might be involved, so looking for mutations genome-wide. And another main approach is the functional approach, which is hypothesis-driven. So an example would be, let's say you are convinced that the serotonin neurotransmitter system is involved in causing autism. Well, then you might hone in on the genes that code for the serotonin transporter or the receptor and look for mutations in patients in those particular genes. The point, though, is that whatever techniques are used, human geneticists hope to come up with a list of candidate genes that are associated with autism, but it doesn't end there. To really nail down genes as being associated with a disorder like autism, you need to do a lot of follow-up work. So, for example, doing mutation screening in large cohorts to see how relevant a gene is to a larger patient population, and also doing functional analysis. So the finding mutations in genes is really just the first step. You need to understand, if you really want to understand how autism comes about, is trying to figure out how those mutations affect the structure and function of a protein. So it seems so unfair to sum this up in just one slide because it's taken many years and research groups to do some of these studies. But so some of the types of techniques, the genome-wide techniques that geneticists use are linkage and genome-wide association studies. So a linkage study shown in the upper left of the slide basically is looking through pedigrees, looking through families, and where a phenotype like autism travels with DNA markers that represent the entire genome. So looking for co-segregation. And more than a dozen genome-wide linkage studies have been conducted in autism. And the running theme is that they often contradict each other. So the reasons for this could be various. So, for example, there are different inclusion criteria from study to study sometimes, different DNA markers that are used across the genome to look for segregation, different statistical packages that have been used, and maybe it's just that there is so much genetic heterogeneity in this disorder. So I really summed it up in this tiny table. If you pick up one of these linkage papers, you find a statistic called a log score, or logarithm of the odds score. So for example, a log score of 3 means that there's 1,000 to 1 odds that the locus or chromosomal region is linked to the phenotype under study. So for autism, linkage peaks have been found on pretty much every chromosome. And here is a summary. For example, significant log scores have been found on the short arm of chromosome 3, chromosome 2, 17, and 7. The other type of genome-wide study that's been done are association studies, and these in a way are even simpler to understand. You basically have two cohorts, affected individuals and control individuals who are typically developing, and you're simply asking, do the cases have more mutations, and in which genes that would be, which chromosomal regions? So a couple of large-scale studies were published a few years back looking at hundreds of patients and thousands of typically developing control individuals. looking for a million or so SNPs, single nucleotide polymorphisms or DNA markers across the entire genome, looking to see are some of these markers overrepresented in the cases versus the controls. And so here's another running theme in that often these genetic studies in autism tend to be low yield. So looking at lots of patients, lots of controls, lots of DNA markers still yields a few results. So this group published a few years back and found that one marker on chromosome 5 reached genome-wide significance. So if you pick up a GWAS paper, you find a figure like this. Often it's called a Manhattan plot because it looks like this Manhattan skyline. So along the x-axis you have the human chromosomes, and on the y-axis you have significance levels. And so this group found one marker on chromosome 5 that reached genome-wide significance for association with autism. It actually didn't even land in a gene. It landed between two genes, cadherin 9 and 10. And the reason why this result was so interesting, besides being one of the first GWAS papers done in autism, was that these genes mediate neuronal cell adhesion. So the thinking being that that sounds like a plausible gene to be associated with autism because it's involved in neuronal development. Okay, so another technique that human geneticists use to understand the genetic contribution for autism is cytogenetics. So this is looking at chromosomal abnormalities, and there's two main types. So looking at balanced abnormalities in which there is no net gain or loss of chromosomal material, and unbalanced in which there is gain or loss of material. So here in this diagram you have two chromosomes A and B which have switched ends for some reason. Either it's inherited or again spontaneous. And then so this represents a balanced translocation, and although there might not be a net gain or loss of material, two breakpoints have been left behind which could disrupt a very important brain-active gene. An example of an unbalanced abnormality is trisomy 21, which everyone is familiar with, and so this involves a whole extra copy of chromosome 21 which could have serious clinical consequences. So I just want to take a second here and talk about karyotypes versus chromosomal microarrays, because I'm sure you've heard of these terms and some might not know what the difference is between them. A karyotype is basically taking a picture of a patient's chromosomes under the microscope after they've been stained with a dye and produce what we call this banding pattern. And a cytogeneticist can actually pick out these chromosomes and pair them up, which I always find miraculous, and then look at the banding patterns to see if there are any abnormalities. A chromosomal microarray involves also looking for duplications and deletions of chromosomal regions. In this technique, you have the patient's DNA, and you also have DNA from a control individual or a pool of control individuals that are stained with different dyes. These are combined together on a fancy glass slide. And so these numerous spots on the slide represent chromosomal regions. And so if you have a spot along the genome in which the patient's DNA is, for example, duplicated, amplified, then that green color will show through. Or if you have an area where the patient's DNA is deleted by a copy number variant, then the control color, the red color, shows through. So this can be scanned, and the results compiled to look for copy number variants throughout the entire genome. And we'll talk about the implications of this in clinical genetic testing in the latter half of the talk. So a number of genome-wide CNV studies have been done in autism. So there was a group a few years ago that found that copy number variants are significantly enriched in certain gene families, certain pathways in autism. So basically these genes, this list of genes that are affected by CNVs are found in maybe one or a couple of families, so they're rare causes of autism. But this group did an important thing, which is take a step back and say, okay, well, this diverse list of genes, are they all found in some common pathway? And so when they did this kind of analysis, they found that a number of genes are, again, coding for neuronal cell adhesion molecules, so involved in neuronal development, which seems plausible in autism. And then another group of genes is involved in the ubiquitin pathway, which is involved in protein recycling. Another group published a paper looking at families affected by autism and found that they have a higher rate of rare genic CNVs, meaning CNVs that affect genes rather than being located in intergenic regions, and this is compared to control individuals, and found their own list of genes. So that's another recurring theme, is that not often do you find two studies that agree on one set of genes. Often you find different genes being highlighted by these studies. And then a recent paper looked at copy number variants in simplex families. So these are families in which there is one affected individual, with the thinking that these types of families are enriched for a spontaneous mutation, since there is one individual rather than multiply affected individuals. And indeed, there were a higher rate of rare de novo CNVs at these chromosomal regions. So again, you might wonder if all of these genes and regions are rare by themselves, found in maybe one or a couple of families, how does this help us understand autism? But the important thing is that they still implicate gene networks. So they still give us an idea about, if we know something about what that gene does, then where in the brain and when in the brain the dysfunction might be coming about, giving us clues about the pathophysiology. So a hot topic in autism and in neurodevelopmental and neuropsychiatric disorders is that there appear to be overlaps in CNVs and gene mutations between different disorders. So we all know that autism and schizophrenia are very different clinically, but it's intriguing that they share some CNVs, shown in this diagram in black. So the question here is, how is that possible? And perhaps that these common CNVs can shed light on each other, on what's going on in autism versus schizophrenia. So I, in my relatively short research career, can still remember the time when we studied one gene in one family at a time and could spend years on a gene, which I've personally done. And then high-throughput sequencing came about, which has really revolutionized and accelerated our research into the genetics of autism. So an example is whole exome sequencing. And so the purpose of this is to obtain the DNA sequence for the entire protein coding region of the genome, known as the exome. And this is just 1% of the genome. So along the chromosome are segments that are genes that code for proteins, and in between these gene segments are intergenic regions that are not known to code for proteins. So here is a picture of this fancy glass slide in which you have 8 microchannels, and you can input the genomic DNA for up to 6 or more individuals in each one of these channels and get the sequence for the entire exome in this one experiment. So now we're talking about going from studying one gene in one family at a time to getting the whole exome, the sequence for 20,000 some genes in about 50 individuals in one experiment. So you can see how much this has accelerated our work. And the power of this comes from the fact that the chemistry allows for the amplification of chunks of the genome so that you can do really detailed sequencing and look for things like rare variants. And so you create these millions and millions of molecular clusters. And the sequencing machine, we have about a dozen of these at our core facility. It sits on a bench top, and it turns out sequence information. So it's really nice to get all this information, but the big challenge is doing the bioinformatics analysis to make sense of all this data. So we get raw sequencing reads, which are stretches of about 75 base pairs or so of DNA sequence. And then we need to align the reads to the human genome reference sequence that we can find online, and then look for variants. Where are the changes? And then, so for just one person, we can get almost 100 million of these short reads. And each nucleotide, each base, is covered on average about 120 times, which gives us confidence that we can find a rare variant. But even for one person, you can find up to 30,000 of these variants. So it can be pretty impossible to go through all of these variants per person. So what to do next is to do some smart filtering to see, well, which of these, or most of these variants are going to be benign. Some of them, maybe just one of them, might be associated with autism. So even if we look at, for example, just novel variants, that's still quite up to 800 or so. So we do filtering by, for example, is the mutation we're finding at a conserved site in the genome? If it is, then that might tell us give us a clue that that site is important for some reason if it's been conserved through evolution. We might look to see what that change does to a protein, whether it changes an amino acid or causes a premature stop codon, meaning truncating the protein prematurely. So we use those types of filters to figure out of all these variants which might actually be associated with a disorder. And another thing we can do is try to look for genes which have been hit recurrently in unrelated families. And that was what was done in two recent large-scale whole exome sequencing studies done in autism. I published a couple of years back. One from the Autism Sequencing Consortium and another from the Simon Simplex collection. And so here is a list of genes. Combined, these studies looked at about 6,400 cases, families affected by autism, and came up with a list of 50 genes that seem to be very strongly associated with autism, meaning that they were hit multiple times in families. But even then, the most any one gene in this list was found affected in families mutated was perhaps a dozen times. So each of these genes are still very rare causes of autism. And they look quite diverse. It just looks like a bunch of acronyms. So how can you possibly make sense of these genes and what they do? Before I go on, I want to mention how valuable these DNA registries are. So these consortiums use, collect and bank DNA from families affected by autism. So it is their participation that really advances in our research and makes it possible. So one of the interesting findings from these exome studies was the fact that a lot of these genes that appear to be mutated in autism and associated with autism are also affected in other disorders. So intellectual disability, schizophrenia, epilepsy. I've already mentioned how there are common C&Vs between autism and schizophrenia, but other disorders like congenital heart disease that don't seem to be on the surface related to autism, but there are some genes that seem to be mutated in both of those disorders. So again, the question becomes like, how can mutations in the same gene lead to such different phenotypes? And that's a hot topic of research. So I want to say that whole genome sequencing is here, which is really exciting. So instead of getting sequence for just 1% of the genome that codes for proteins, we can get, now it's becoming more and more affordable to get the sequence for the entire genome, which is 3 billion base pairs. So all that bioinformatics challenge I mentioned under a whole exome sequencing, you can multiply by 100. And so instead of getting 30,000 variants, we'd be getting 100 times that in per individual. So here is an example of a whole genome sequencer that again can fit right on a bench top, and the fancy glass side in which you input the patient's DNA and you feed it into this machine, and now in a matter of days you can get the sequence for the entire genome. So back in 2001, when the whole genome, the Human Genome Project was completed, it cost $100 million to sequence one human genome, and it took many years. It was the effort of numerous labs and many researchers, and now in 2015, we're very close to the $1,000 genome, and instead of years, it can take a matter of days. So again, really accelerating our research. And if you know Moore's Law, it basically says that computing power doubles about every 18 months or so. So if your technology is beating Moore's Law, then you know it's advancing pretty rapidly. And that's definitely true, quite dramatic for human genome sequencing. So now that it's becoming faster and more affordable, more and more labs are doing whole genome sequencing in autism for themselves. So papers are now starting to trickle out. There were a couple of papers published in 2013. One group looked at 32 families, found a percentage that had de novo mutations and interesting genes, and a third that had inherited mutations, and came up with a gene list like you've seen before, a variety of genes often active in the brain, different neuronal processes. Another group did whole genome sequencing in 10 members of one extended family, including 2 members that were affected by autism, and found that this pair of affected individuals shared a mutation in ANC3, which is expressed in the brain. And another group recently published a whole genome sequencing study looking at 85 quads. So these are families with 2 unaffected biological parents and 2 affected children, and found this interesting result that 70%, so the majority of the affected SIBs carried actually different autism-relevant mutations. So they didn't even always share the same mutation that could be causing their autism. So just again speaking to the high degree of genetic heterogeneity in this disorder. And these SIBs that were carrying the different mutations were also happened to be more clinically variable, which sounds logical. So these papers, as I said, are now being published, these studies in whole genome sequencing, and there are projects underway as we speak with large-scale whole genome sequencing looking at hundreds of families, a few thousand families. Autism Speaks itself is sponsoring the Missing Project, which has a goal of doing whole genome sequencing in thousands of families affected by autism. All right, so I like this slide from the Emory Genetics Laboratory looking at the genetic architecture of an autism and how much heterogeneity there is. So there are definitely a subset of the cases of autism excuse me, that are affected by single gene disorders, like those defined genetic syndromes that I showed before. Also, there are a subset of cases that have cytogenetic abnormalities that have been published in the literature. And then there's an entire large subset of cases of autism that seem to be due not to these single gene disorders or cytogenetic abnormalities, but to multiple genes, each contributing a small amount of risk, possibly also involving environmental triggers and factors. So the genetic architecture of autism is quite variable. So again, how do we make sense of this diverse list of candidate genes that we're drawing up now because of all these high throughput sequencing approaches? Well, I believe that developmental neurobiology is really going to help us in this way to try to inform autism pathophysiology. So I wanted to highlight 2 studies that were done. One of them here at Yale, this one by Kang, where basically the gene expression profile of the human brain was mapped. So in this study, a couple of postmortem brains were taken, tissue was sampled at numerous spots throughout the brains, and the genes that were active in those spots were mapped. So again, creating this transcriptome map of the human brain. And in this study, numerous brains, postmortem brain tissue was examined, again looking for when genes are being turned on and off, when they're active in the brain throughout the lifespan. So on the x-axis of this graph, you have the age of the postmortem brain ranging from fetal stages all the way to 80-year-old brains. On the y-axis is the signal intensity of the gene expression, so anything above 6 is considered positive. And neuroligin 4X is a gene strongly associated with autism, and you can see that it's highly expressed throughout the lifespan in numerous brain regions, including the neocortex, the hippocampus, the amygdala, etc. So one thing we can do with a diverse gene list is to look and do co-expression analysis. And this means looking to see if the expression profiles of these candidate genes are similar. In other words, do they turn on and off at the same time throughout the lifespan in various regions of the brain. And the reason to do this is because we can think that if 2 genes are being strongly expressed with each other, their expression is coordinated, then perhaps that means that they are acting together in a certain molecular or cellular pathway. Again, giving us ideas that perhaps that would be the site of dysfunction in autism to understand the disease mechanism. So a really interesting study was published a couple of years back doing this type of gene co-expression analysis where genes associated with autism, because they were identified as having de novo mutations in simplex families. This data set was intersected with the human brain transcriptome data set, in other words, where genes are being turned on and off and where and when in the brain. And a co-expression network analysis was conducted and found that a significant number of autism-associated genes are indeed expressed together. Their expression seems to be strongly coordinated. And not only that, this is happening in mid-fetal development, giving us clues as to when there might be the disorder might be unfolding, and in the prefrontal and primary motor somatosensory cortex. Not only that, but specifically in layer 5 to 6 of the cortex and glutamatergic projection neurons. So this was a very important study because it took a list of autism-associated genes and was able to drill down to where and when in the brain the dysfunction might be arising. So that's the power of doing something like gene co-expression analysis. Another really hot topic in the field is to do stem cell modeling, to kind of do that follow-up work of examining mutations in proteins and trying to figure out how does that affect a protein structure and function and cellular processes. So, you know, the big challenge in neuropsychiatry is that we can't go around getting brain biopsies for study from our patients. But this technique that actually won the Nobel Prize in 2008 has really helped our field because we can take patient cells, so these can be hair cells, they can be blood cells or skin cells, and they can be reprogrammed by adding transcription factors and reprogrammed into an immature form, induced pluripotent or iPSC cells. And the power of this is that these immature cells can be, again, differentiated into any type of cell of the body. And of course, we're interested in neural cells. And so we can really create kind of a disease model in a dish, looking at neurons from a patient with autism with its entire genetic background to study whether a mutation affects, for example, neuronal differentiation, synaptic development, morphology, et cetera, and not only that, look to see what abnormal phenotypes we might be able to find, but then serve as a platform for example, drug testing, high-throughput drug testing. So this is a really exciting development for autism and for neuropsychiatry in general, because instead of trying to study gene mutations in blood cells, which are relatively easy to obtain, we can now actually look to see what these mutations are doing in neuronal cells in a dish. So I wanted to shift gears a bit from recent advances in autism genetics research and talk about clinical genetics testing. I know there's a lot of interest in this. So why do clinical genetics testing? There's a number of important reasons. One being that you might be able to identify a known genetic syndrome. And the reason this is so important is because there is often additional organ involvement. So for example, in individuals affected by Fragile X, there's often connective tissue problems. In tuberous sclerosis, there can be problems with other organs such as the heart, the kidney, and the lung. So identifying such a syndrome can allow for anticipatory guidance and treatment. There are important implications for genetic counseling. So whether a mutation is inherited, like in this pedigree, or de novo, a spontaneous, can have implications for a couple who are thinking about having another child, and what is the risk for the subsequent children to have autism, and also for aunts and uncles who are worried if there is a mutation being passed around in the family. Clinical genetic testings can be, and the results can be helpful in obtaining needed services for an individual, and also to avoid pursuing further diagnostic testing, which can be very expensive and time-consuming. So the various academic societies have guidelines about clinical genetics evaluation in autism. The American Academy of Pediatrics published a clinical report that was recently reaffirmed a couple of years ago. So it suggests that karyotype and Fragile X testing are reasonable in autism, especially if there is intellectual disability. A clinical report in 2014 looked at clinical genetics testing in intellectual disability and global developmental delay, and recommended, instead of karyotypes, looking at chromosomal microarrays. And this is why I wanted to show you that slide explaining the difference between the two, because it turns out that CMAs are higher yield than karyotypes, and also Fragile X testing. The American Academy of Child and Adolescent Psychiatry published practice parameters that suggest genetic testing can include karyotype, a CMA, or Fragile X testing. The American Academy of Neurology reaffirmed its guidelines in 2014, recommended a karyotype and Fragile X testing if there's intellectual disability, a family history of ID, or dysmorphisms. And then the American College of Medical Genetics has practice guidelines that I feel are the most detailed and provides a tiered evaluation to try to decide what clinical genetic testing is appropriate. So in the first tier, the ACMG recommends that all families have a 3-generation pedigree documented to try to help discover if there is something that's inherited or spontaneous, or de novo, I should say, in the family. To identify known syndromes or associated conditions with an examination with special attention to dysmorphisms. If a specific syndrome is suspected, to do that targeted testing. And if clinical indicators are present, then to do metabolic and mitochondrial testing. So in other words, that type of testing is not considered an automatic test to do because they tend to be relatively low yield. But if there are indicators present from the history or the physical examination, then it is reasonable to do metabolic and mitochondrial testing. The ACMG also recommends a chromosomal microarray in all individuals on the spectrum, and then DNA testing for Fragile X because there is this significant overlap between Fragile X and autism. So routinely in males, and in females if there are indicators present from the family history or the phenotype. If all of these clinical genetics tests are negative from the first tier, one can consider a second tier of tests, which includes more specific gene testing. So MECP2 is the cause of the vast majority of cases of Rett syndrome. And so that can be recommended for females with autism because there are anecdotal reports of MECP2 mutations in girls with autism but who do not have Rett syndrome. MECP2 gene duplication testing in males if the phenotype is suggestive because there is evidence for this in the literature. P10 testing, so this is a gene involved in overgrowth syndrome. So in a child with macrocephaly, you can consider looking for mutations in the P10 gene. And then a brain MRI, again, is not an automatic test, but really a test for if there are specific indicators. So, for example, microcephaly or regression, seizures, etc. So the ACMG projects these diagnostic yields. So for chromosomal microarray, 10% of the time, they expect that you would find a positive result. 1 to 5% of the time for Fragile X testing, 4% in girls for MECP2 mutations, and so on. They estimate that if you go through these tiers, these clinical genetic tests, that you might be able to identify an etiology or a positive result in about 30 to 40% of individuals on the spectrum. So that's a good proportion of cases. So whole exome and whole genome sequencing are also, or at least whole exome sequencing is now available clinically. So what are the indications for this? And so the ACMG published a policy statement in 2012. I haven't seen an updated one. That it's indicated when there's a strong implication of a genetic etiology in a patient, but the phenotype doesn't correspond to any specific disorder that can be tested with a clinical genetic test. Also, if there is a defined genetic disorder with a high degree of genetic heterogeneity, so a number of genes that might be causative. In this case, it's more cost-effective to do whole exome sequencing than look at each specific gene. Because the price for whole exome sequencing has also dropped dramatically as it has for whole genome sequencing. Also, another indication is a likely genetic disorder, but specific genetic tests are negative. So, for example, my research group was examining a patient with alternating hemiplegia of childhood and didn't find a mutation in the known gene. And so doing whole exome sequencing enabled us to just survey all the genes to see if there is a new candidate gene for that particular disorder. And then a fetus with a likely genetic disorder, but again, where specific genetic tests are negative to look for mutations in other genes, can also be an indication for whole exome or whole genome sequencing. So a clinical genetics evaluation really should be routine in the medical assessment of autism, and this regardless of symptom severity. So I'm often asked by clinicians, my clinical colleagues, if they should reserve clinical genetics testing for those kids on the spectrum who have dysmorphisms and are particularly severe. So this group published a paper in 2003 trying to address this question and looked at patients with autism and patients with a milder form called PDD-NOS, and looked to see the rate of an abnormal genetics test. And so even though the numbers are on the small side, they found that the rates are pretty comparable. And one of the C&V studies found that low IQ is actually not a predictor of the presence of a copy number variant in at least simplex families. The bottom line being that everyone on the spectrum really does deserve some clinical genetics testing, and not just those on the severe end of the spectrum with dysmorphisms. So without an identifiable etiology, genetic counselors can use empiric recurrence risk data. So this is again from the ACMG practice guidelines. So for full siblings, the recurrence risk for another child is about 3 to 10%. As I mentioned, there are papers showing even higher rates for the sibling recurrence risk. If the affected child is a girl, the recurrence risk is 7% and only 4% for a boy. And the reason for this discrepancy is the feeling is that for girls who are affected by autism, and you know there is a skewed male to female ratio in autism, that it takes a greater genetic hit, meaning a larger CNV or more genes affected or a more deleterious mutation for a girl to have autism. So perhaps that girls have a protective factor that takes a larger genetic hit for a girl to have autism. So this is another hot topic of research as to what these protective factors might be, if there are such protective factors. So that if you do have a girl affected by autism, that the recurrence risk goes up because there's a larger genetic hit in that family. And then if there are multiply affected children in a family, the recurrence risk really jumps up to 33 to up to 50%. So you may have come across these commercial autism gene panels. This is just one example I found on the web. And so these panels are often kind of pitched to families, to parents who already have a child with autism, and they're very worried about a second child that might have autism. And these commercial gene panels are meant to try to estimate risk in a second child for autism. So I would just be cautious about these panels. They can be quite expensive. I'm not clear if they are covered by insurance, but the thing is, when I look at some of these gene lists included in these panels, there are almost all of these genes are rare, again, rare causes of autism. So if a gene has been associated with autism in the literature and it's been found only in 1 or 2 families, you can imagine that these gene panels are going to be on the low yield side. And so to the credit of this particular company, they at least say, I've highlighted in the red box, that while researchers have identified a number of genes which may likely play a role in the development of autism, most genes which have been shown to be associated with autism are represented in less than 1% of individuals, in other words, being rare causes of autism. There is no one gene or chromosomal region that explains any substantial proportion of individuals with autism. So, but there are definitely examples of these commercial gene panels, again, that cost thousands of dollars and have some genes that are weakly associated with autism or are rare causes of autism and are not as explicit on their websites. So that's why one should proceed with caution with these commercial gene panels. So another common question I get, especially from parents, is, participating in genetics research, is that important? How will that help their child? And the thing is, at this moment, it might very well not help a particular individual to participate in genetics research studies, but I have a couple of really good examples of why it's so critical to do this research, why it's so important for the larger research effort. So going from genes to pathophysiology to treatment, one example is Rett syndrome, which I mentioned before is due to mutations in a specific gene called MECP2. So MECP2 is one of these master regulatory genes because it controls the expression of other genes. And patients with Rett syndrome show abnormal neurons, but not neuronal death. So the important question becomes, can viable but defective neurons be repaired? A research group developed a mutant mouse model lacking this gene, and they developed neurological symptoms similar to what is found in human patients. They genetically reactivated the gene in adult mice, and that's important here. So they did not do this prenatally or in pups. They did it in adult mice and were able to reverse some symptoms, including deficits in neuronal signaling in the hippocampus, which is a part of the brain that's been associated with autism. So I think this tells us that there's a lot more plasticity to the brain than we always suspect, and that the absence of MECP2 didn't irreversibly damage neurons, that it was still possible, even in adult mice, to reverse some of the symptoms. So that hope is not lost, even if we believe that these genes are working in mid-fetal stages in utero to start developing autism. Another example is Fragile X. Loss of FMR1 gene causes Fragile X syndrome. It's like MECP2 in controlling the expression of other genes. Because scientists were able to find the genetic etiology of Fragile X, they were also able to determine that the pathophysiology involves hyperactivity of a glutamate receptor called mGluR5. So a research group developed, again, mutant mice lacking this gene, which developed neurological symptoms similar to what is found in human patients with Fragile X. And so they reduced expression of this hyperactive glutamate receptor by half and showed rescue of symptoms, including alterations in dendrites and hippocampal protein synthesis. And then furthermore, the GABA agonist arbaclofen, which is a pharmaceutical compound on the market with a lot of safety data already in use, was found to correct increased synaptic protein synthesis and spine density in mutant mice. So again, we're going from a specific gene. Once we have pinpointed that, we can really uncover the disease mechanism and try to find targeted treatments. Targeted treatments for these disorders. And that's why it's so important to pursue genetics in a disorder like autism. So I wanted to end with some future directions for the field that I think are really important. One is that we really must increase our study populations. So other complex disorders like diabetes and hypertension, those papers study tens of thousands, 50,000, 60,000 patients. And maybe the reason why we haven't uncovered all of the genetics of autism is because the largest scale studies are still a few thousand. And we really need to increase our study populations. I mentioned whole genome sequencing, and so getting the sequence of the entire genome and not just the genes. So that enables us to look at the sequences of regulatory elements, mitochondrial sequences that you don't get from whole exome sequencing, because perhaps these are sources of genetic mutations as well in autism. I mentioned pathway analysis, how when you have a list of diverse genes, to make sense of it is to perhaps take a step back again and look to see are certain gene families involved, or certain molecular or cellular pathways involved, to try to give us more clues about the disease mechanism. I didn't touch so much on epigenetics, and these are chemical modifications of the DNA sequence that affect gene expression. This is a challenge for our field because usually epigenetics is very tissue specific, and as I mentioned, we can't go around getting brain biopsies from our patients. But this could also be a source of problems, contribute to the genetic etiology of autism that we haven't really uncovered yet and comprehensively surveyed. Looking for biomarkers is another hot topic, from things like neuroimaging and eye tracking, EEGs. So even with gold standard diagnostic instruments, diagnosing autism is still very subjective. So try to reduce some of the heterogeneity in the study populations. We can look for biomarkers that will make the subsets of the study subjects at least more phenotypically homogeneous, so more objective measures. I mentioned the genetic overlap between different neuropsychiatric disorders, so they can shed light on each other. And really, I find that the holy grail right now is that we're, finding a list of genes and mutations that are strongly associated with autism, but really that's just the tip of the iceberg. We really need to do the functional analysis of variants to bridge genetics and neuroscience, so doing those follow-up in vitro and in vivo studies, looking at postmortem brains, looking at those stem cells that I mentioned, iPSCs, and animal model systems, to try to uncover how precisely do mutations in genes cause disruptions in protein structure and function. And then, although I said that commercial diagnostic autism gene panels are perhaps a bit premature right now, we would love to have diagnostic gene panels eventually as we gain more confidence about the genetics of autism. Okay, that's the end of my presentation. Thank you for your attention, and I'll welcome any questions.
Let's see, so we have a few minutes for some questions. Let me see, some people have submitted questions via the questions panel. So one of our parents who's on the call has said that she has Asperger's and has a son with autism and a daughter who's not on the spectrum. Would you suggest that the daughter get some of these tests done before she tries to have children to see what her chances are of having a child on the spectrum?
Sure, and the ACMG, the various academic societies would recommend that to see if there are mutations which might have been passed along to the unaffected daughter. So yes, I think that would very well be recommended and justified to do that genetic testing.
So that's the only question that we have right now. We do have a few more minutes if folks have other questions that they want to ask.
I think that was just a wonderful presentation, Dr. Gupta. Thanks very much. I appreciate at the end your discussions of how some of the different ways of looking at some of the genes and their activities is where things seem to be headed at this point, and how that may help us identify some novel treatments in the future. Some of the animal research is very exciting, as you were pointing out. One of the frustrating things is that humans don't always act like laboratory mice. So your example with the mGluR5, which was extremely promising in the mouse model, but when we translated it to humans, it is not held up as well as we had hoped. Nonetheless, there are a number of others that are still moving forward along this line, and I think it's a very exciting area to see this when you see the really robust effects they have in animal models. The hopes that it could happen in humans just is one of the things that keeps me excited about coming to work every day.
Yeah, and obviously there's a big leap between mice and humans, but those studies were very important, at least as proof of principle, that neurons are not irreversibly damaged, and perhaps there are phenotypes that can be rescued even later in life, so that it's not hopeless. So again, a lot of work to get to humans, and Mother Nature is always more complicated than we think, but those still were offered a lot of excitement, those studies.
Any other questions from our audience, Audrey?
Yeah, so we do have another question from another parent who has a 3-year-old with autism, and if there's any way to determine which parent could be potentially responsible for any corrupted genes that may have led to autism.
So the clinical genetics evaluation would start with examining the affected child first to see if there are any mutations that could be associated with her condition. And then if there is one, then typically what we do next is test the parents to see if it is a mutation that is spontaneous in the child versus inherited from the parents. And so that would require testing both the mother and the father, the biological parents, to see if there was anything passed along.
Great, thank you. So that's the end of our time, and we don't have any questions. Oops, we don't have any questions, any additional questions. So I'll just again thank Dr. Gupta for her presentation, and just remind everyone that CME credit will be available, and everyone will be receiving a recording of today's session with an evaluation form. And if there are any other questions that you'd like to pose to Dr. Gupta, you can email them to me and I can pass them along. So thanks everyone for joining today.
Great, thank you.
Thanks, folks.
Thanks.
Bye now.